Thank you. Okay, wow. Well, greetings, everybody. I apologize for the technical delays, and I want to thank Kim for her patience. It's always important to have someone very calm in audiovisual to allow us to get the job done. I have many people to thank, and I have to thank Prof. Chung for being such a good friend during so many years, and I will show some slides regarding that. I'm very honored to be here in person and delighted to be here in person for this incredibly important celebration of 20 years of the faculty and the establishment of UTAR. I'm also very grateful to Prof. Ong, the organizing committee, Dr. Lee, Prof. Lee, for introducing me, and I hope I can entertain you for 45 minutes or so and give you some new ideas and new thoughts about cell and gene-based therapeutics in blood disorders, which is my topic for today. And obviously keeping in mind aspects of stem cells and cancer research, which are part of what I do. But I want to give a broader outline as well to give you some insights as beyond that as well. So here are my disclosures. I have a number of different companies that I've worked with over several years, and importantly, I serve the Australian government as chair of the Gene Technology Technical Advisory Committee for the Office of the Gene Technology Regulator, now for more than 15 years as its chair. I just emphasize that I don't speak on behalf of the Australian government when I'm talking about regulatory affairs. Before I launch into my topic of interest today, I wanted to say something that I haven't ever said before, even though I'm coming here for almost a decade, and that is to reflect on the people who lived in my country for more than 60,000 years, perhaps 75,000 years. That's the Indigenous Australians who are currently very much in the news as Australia struggles with its colonial past from England, as we've all seen back-to-back a mourning for the Queen's passing and the new King. Australia, of course, shares with Malaya and Malaysia a history of colonialism, and one that we struggle with still to this day due to the trouble that invasions and past historical events have caused all of us and our families. And so it's important to recognize that the place I come from has also struggled with that. And the Australian Indigenous people who have survived in one of the most harshest environments in the world, in Australia, a very large country, but mostly desert, represent perhaps the oldest continuous culture on Earth, with rock paintings that you can see in the background there dating back by carbon dating more than 60,000 years. And that culture is an inanimate, in that culture, even the inanimate world, the rocks, the sea, the mountains, the air is alive. Animals, plants, and natural forces are all energized by a spirit. And for those from a TCM background, I think that might resonate. We humans are on an equal footing with nature, and we are part of nature, and we are morally obligated to treat animals, plants, and landforms with respect. And that's a culture that we share coming from Australia. So also reflecting on the past, but the relatively recent past, I had to celebrate the fact that even though it's 20 years for UTAR, it's almost 10 years since I've been coming almost every year, aside from COVID, to this beautiful campus and to this glorious university, which has grown in stature and relevance both in Malaysia and beyond. I was first invited to speak here at Bandar Sungai Long in 2013, and I think I came twice that year, SK, in May and then later in December that year. But I was very privileged and humbled to be appointed as an adjunct professor in 2014 to UTAR, where I have remained every year in that role. And there are just two of the recent conferences in 2017, and the most recent one that I was at before today, which is in 2019. I had the great delight with Prof. Ong and Prof. Vera to visit the new Kampar Hospital, both the TCM hospital and the nearly finished beautiful hospital. And I have to thank Prof. Ong. Thank you, Alan, for these beautiful photographs and some that were taken with my phone. Alan's ones are much better, but really enjoying the campus that many of you won't have even been to yet. But I had the great privilege to go there last Thursday and visit and explore the new building with Madam Chu, who was a wonderful leader and developing things. So I'm just reflecting on the fact that the official launch of UTAR was the 13th of August 2002. We only 1 month short, further than the 20 years. And it made me think about a 2-decade range. And that's really where I'm going to start the substantive aspect of my speech. And I want you to now almost close your eyes and ask you a question that I want you to think about while the slide is black. And that is, what do you think about when you think about GMOs? Do you think about genetically modified organisms? What ideas come into your mind straight away when you hear the word GMO? As a person, as a scientist, as a physician, GMO, what do you think of? Well, whatever it is that you think of, this is what the public think about, okay? They think about scary things. They think about frankenfoods and naughty, destructive plants, things that will take over the world, things they should be scared about, things that might damage them or their children or their families, foods that could be poisonous or destructive or injurious to their health. And these are just some of the posters and the slides that I've collected over a number of years that have been put into the press by people who are not in favor or scared of genetically modified organisms. And even at their best, if you talk to your colleagues or your friends who are educated about science and technology, what do they think about when they think about GMOs? Well, they may not think about such scary things like this, but they certainly think about technologies that we should be worried about, you know, crazy people standing in a laboratory who are doing crazy things that we should be a little bit suspicious or kind of worried about, that it's not quite right. And so as we enter, and as I will highlight throughout my talk, the important next phase of medicine, namely involving genes and cells, I would put it to you that the dawn of molecular medicine is one where society needs to deal with really important topics, because the technical challenges that most of us in this room and others in the world struggle with on a daily basis to get everything to work, to overcome the problems and the challenges of technical mastery, that's just in that little black circle in the middle. That's just the science, okay? But the public, they say, oh, let the boffins, let the scientists worry about that. We're worried about much bigger issues. And so from the public area, we are the ones who are the outliers. We are the ones who are outside. We're the ones who aren't thinking straight. We're just dealing with the science. But politicians, regulators, the general public, lawyers, philosophers, ethicists, these are the people who need to deal with all of the other issues that are present in this bubble of different things going on. So I want to reflect on that as Malaysia enters a new era of genetically modified organisms. Now, it turns out, I looked it up only yesterday, Malaysia has already approved 8 different GMOs, but these are all plant GMOs. There are no human genetically modified technologies that have been formally approved for regulatory compliance within the context of routine medicine or prescription paid for by the government. But this was a paper published partly by authors from Malaysia about the NPRA in Malaysia taking their first steps in 2016, and then the mandatory registration and enforcements were scheduled to begin nearly what is it now, more than 18 months ago. But SK reminded me last night at dinner that they still have not been promulgated, and these regulations, I think, are not yet enforced in terms of cell and gene therapy regulation. So we still wait for that regulatory approval and structure to be enforced. But the reason why I highlight all of that history is because I can reflect on 20 years now of experience in regulating genetically modified organisms. So in the year 2000, the Australian government created the Gene Technology Act, and it was a very ambitious and broad act. And this act was important because it comprised not just plant GMOs like GMOs, sorghum or maize or wheat or barley or any of the other commonly used GMOs like cotton, but it also, under the same legislative framework, regulated human genetically modified organisms and animal genetically modified organisms, and indeed all bacterial and so on. So therefore, one law covered everything, all recombinant DNA technologies. And in many of these slides, I have given you QR codes. So if you want to hold your camera up to the QR code, you can actually go straight to the paper that was published. And you can see that this was a publication only earlier this year from a group of us who were reflecting on the 20 years of a stable regulatory legislative process that has criminal consequences if you fail to comply in terms of that law. It provides a science-based assessment of the potential risks of gene technology for health and safety of people and the environment, and the reforms occur every 2 or 3 years legislated so that new technologies can be absorbed. Because, hey, in the year 2000, we had no idea about CRISPR-Cas9. It was just a twinkle in somebody's eye, even back then. And so all of these new technologies like RNA-based therapies, the vaccines that we currently have, none of those things were properly available, or indeed even some of them imagined back then. And yet the law is robust enough to anticipate these changes and even though the act has not been changed, regulations interpreting the act can absorb the new changes. And the reason why that's so important is that there are so many people worldwide who are not paying attention to the potential risks and consequences and ignoring the public's concern about using GMOs. And in this room, I have spoken a number of times in the past about some of these publications from 2016, 2017, highlighting the fact that even if you do no harm with GMOs or cells that are unproven, at a minimum, you are compromising a patient's opportunity to get something that does work. And as physicians, we all have an obligation to do no harm, but more importantly, we have an obligation to warrant the trust of our patients. And if we are giving or prescribing or using treatments that are not working, then that is just as bad as giving something that's damaging, because in most cases, there is something that is available that at least will help and has been proven to help using empirical medicine. And so that has been highlighted in a book that I published relatively recently with Dr. Carl Power, hopefully shown on this slide here, that describes some of the history of cell-based therapeutics, starting from cells in bone marrow transplantation and then moving towards some of the more recent technologies and highlighting some of the charlatans and fraudsters in that area. So talking about the Australian regulatory system by way of giving you some insights into what Malaysia may be considering going forward, our system is based on having one individual known as the Gene Technology Regulator. And that person is obligated by law to properly regulate all GMOs in Australia, plants, animals, and human gene therapies. And these are the highlight features of the legislative structure. And as the chair of the main advisory committee to the regulator, which is called GTAC, the Gene Technology Technical Advisory Committee, I have to try now through 3 different regulators. They've changed over the years. I've outlasted 3 different regulators I've been on the committee since its inception, but I've been the chair for now 15 years. And these are the features that one has to juggle. And I highlight in just one example at the very bottom of the list, conflicts of interest, because by virtue of the fact there are 20 professors on the committee, some of them are plant experts, toxicology, environmental, physicians, immunologists, vaccine experts. All of these people come together, some of them bringing commercial expertise and potentially pursuing their own research. So we have to manage conflicts of interest by declaration and transparency. And that's just not the only regulatory apparatus that we have available, because the OGTR, the Office of the Gene Technology Regulator, interacts with a number of other regulatory bodies in Australia. And importantly, towards the top right there, the Therapeutic Goods Administration, which is responsible for regulating all medicinal products in Australia and providing their approval. But also we have human research ethics committees locally. We have the Environment Minister and local government authorities within the states and territories. We have the gene, whoops, sorry, I've done something wrong there. Apologies. I am screen sharing, but something's gone wrong. It's okay. Something's gone wrong.
The connection.
Is it the connection? Yeah, it could be the connection. No, I have Wi-Fi. I think I have to start again, yeah?
Yeah.
What am I going to do? Share?
Yes.
Yes, share. This one again. Yeah. Oh.
I think I have to open the power of the key.
Yeah. Oh, that should work.
Yes.
There we go. Got it. Sorry about that. So all the GMO licenses require Institutional Biosafety Committee, Therapeutic Good Administration, and Human Research Ethics Committee consideration, and they all interact with the Gene Technology Regulator. And so, of course, as the chair, I've been involved in this committee for many, many years. You can see all of the different committees, and most recently, of course, because of COVID, everything shifted to Zoom conferences, just like this hybrid meeting is. And as Prof Chung said earlier, the future of meetings will all be hybrid, just like this one is. And I commend you on realizing that today. So I have published now several papers in the last 6 months regarding the history of gene technology. And it's not my intention to go through this history, but you can look up these papers. They are part of the QR codes that I've shown and will continue to show. But this basically highlights that there have been a series of technical and scientific developments over the last 50 years that have allowed us to achieve and realize the dream of using genes as therapeutics, both living therapies in cells, because of course, genes can't work outside of a living environment because they are simply information encoding instructions to a cell as to how to produce a protein. So for me, always since I've been studying gene therapy, it is in marriage with cellular therapy, because genes can't work outside of a cellular context. And so we can do gene therapy in vivo or gene therapy ex vivo, and I'll give you the briefest examples. But this history basically shows you that as time has gone on, there have been a series of technical developments that have allowed us to achieve therapeutic gene technology and cellular therapies. And some of the commercial successes have been highlighted there in brown towards the right-hand side of the scale. Now, my reason for showing this is not to go through all of the details of these scientific and technical developments, but to show you that during the same time frame of 50 years, there have been a series of regulatory events worldwide that have developed in partnership with the science and technology. They have responded to and tried to anticipate some of those scientific and technical developments. And, you know, many people, young people in the audience and the students listening, won't remember that back there was a conference in Asilomar in California when the first recombinant DNA technology was discovered and developed. And that Asilomar conference said, we think that the world is not ready to do recombinant DNA technology. We're going to put a moratorium or a pause on that technology before we actually begin. And there was 2 or 3 years when GMOs were essentially banned internationally. They couldn't proceed until they were confident under very strict controls that gene technology was safe to proceed, because there were people who were saying this might be the end of humanity, the end of all life on the planet. We're all going to be converted into a grey goo. It was just at the same time as the Cold War. You know, SK, you'll remember, it was like there was going to be nuclear Armageddon, or we were going to die from some kind of virus that was going to take over the world, or some recombinant DNA technology that was going to absorb all of life and turn it into a grey goo. And so all of those technical developments are reflected by regulatory developments to make sure that our patients, as well as the population in general, can be assured of the safety of GMO technologies in plants, in foods, and in therapies. And what I've also highlighted on the bottom, along the base of the regulatory events, are the actual regulatory committees and institutions in Australia, at least, that have been involved in pursuing those regulatory constraints. And these are the developments in terms of regulatory reforms over the years. Over 20 years now, we've had a first, second, and third update of reforms, which I won't go into, but these are the anticipated adaptations of the law, which have allowed us now to for example, go into site-directed nucleases and guide nucleases using technologies that involve short RNA molecules and vaccines and so on. And indeed, earlier this year, Australia was the second country in the world in a committee that I was involved in with 4 scientists, embryologists, in mitochondrial donation. So now we have legislated approvals to proceed with mitochondrial donation to allow families who are affected by this devastating diseases to take advantage of reproductive technologies using mitochondrial donation, otherwise called 3-parent babies. So just briefly finishing up on regulation of gene technology, this really tells an incredible story. It's 2 simple lines, but it shows how the committee of GTAC and the gene technology regulator in Australia over these different timeframes has dramatically seen an increase in human gene therapy clinical trials, whilst there's been a general decrease in the applications for plant-based technologies in terms of licenses to deal with GMOs. And these are the clinical GMOs in terms of clinical trials over the years. You can see a significant increase in clinical trials using GMOs. And this on the bottom there shows the commercial therapeutic approvals, including Luxturna for RPE65 retinitis pigmentosa, Zolgensma for spinal muscular atrophy, and of course the CAR T-cell therapies, Kymriah and Yescarta for acute lymphoblastic leukemia and diffuse large B-cell lymphoma and other emerging lymphomas going forward, as well as now most recently multiple myeloma. The parent organisms are represented here in terms of the Australian approvals, and mostly adeno-associated virus has been the most commonly approved viral vector system to deliver gene therapeutics. But you can see that adenoviruses in cancer, as well as other viruses used for vaccination, or oncolytic viruses used in cancer therapeutics, all have a place in GMO technologies. And so these are the examples of gene-based therapies now approved in Australia. I won't allow myself sufficient time to go through all of them, but as you scan down, you can see all of the different categories of gene technologies are mentioned there that I just gave mention to. And importantly, the COVID-19 virus vaccines are listed down in the bottom there, using a revolutionary new RNA-based technology and liposomal technology to allow and facilitate the availability of these revolutionary vaccines that will allow us now to proceed with much more agile vaccine approvals that shorten the time to regulatory approval based on these technologies. And again, the regulatory approvals also contract down to institutions. And I hope many of you who are familiar with these technologies are aware of the institutional requirements and regulatory compliances available within your institutions. And I would encourage all of you to consider joining your equivalent of institutional biosafety committees to contribute to the regulatory requirements. And so this is just a poster with the executive officer of our Institutional Biosafety Committee, Dr. Gabrielle O'Sullivan, and a poster presentation. This was my first international trip in almost 3 years to ISCT, where many of you know I was the ex-president, and that was in San Francisco this year, which was very enjoyable. So all of these things can be summarized and highlighted in the phrase that I've extracted from one of these two papers that we published this year. And that is, even though gene technology has been advertised and expected for decades, only now are we really realizing true clinical implementation of gene-based technologies and those going into cells. So many technical and regulatory hurdles have been overcome, but we have a long way to go before they become routine in the practice of most physicians. But as new technologies are being accepted, these have changed the way we do medicine in some aspects within the clinical context. And the main areas where these have made a difference are rare diseases and oncology. These are the dominant areas where GMOs have been accepted. And this highlights the gene technology in the USA especially, but also worldwide, where they are the most targeted therapeutic areas, including RNA, gene, and non-GM cell therapies. This really addresses what is one of the great unmet needs for medicine in the world, and that is rare diseases. Now, I want you to just think for a second about when I talk about rare diseases, most people go, well, that's esoteric. That's something that we don't need to worry about because it's rare. I mean, I'm worried about cardiovascular disease. I'm worried about cancer. I'm worried about Alzheimer's. I'm worried about diabetes, because the burden of disease of these common diseases is very high. But I want you to think for a second. Rare diseases are rare because they occur only one in 5,000 or something like that. It's a rare disease. So individually, they are rare diseases. But collectively, if you add them all up, they are very, very common, because there are some 7,000 known rare diseases worldwide, of which 80% have a genetic origin. All right, thalassemia is not that rare, but it's certainly a genetic disease with an origin and very affecting a large number of different individuals. 80% of these rare diseases are genetic in origin, and yet only 5 or less % has a therapy available that is specific to it. So this is an incredible unmet need globally. And to my mind, the only way we can address these diseases with a genetic root cause that is rare individually but common collectively is using gene technology. And so as we go forward in the coming decades and adapt RNA-based technologies, CRISPR-Cas9 technologies, gene editing technologies, and addition technologies that are now just emerging, I think that we will be addressing the unmet needs of individuals with rare diseases, which are collectively a massive burden and an unmet addressed issue. We do have 20 years of approved gene therapies worldwide, and the first one in the world was actually in China, which is an oncolytic virus called Gendison from a company that is called Sibiono, and there have been a large number of these technologies approved subsequently, and there is a list that you can scan briefly. And now the dominant type, of course, is CAR T-based therapeutics for various blood cancers, in particular B-cell malignancies. Even in the first quarter of 2022, in other words, this year, we have new CAR T-cell based approvals in the USA for multiple myeloma and new approvals for hemophilia and thalassemia. And just in our own institution, this is a summary of the history of how we implemented CAR T-cell based therapeutics. We participated in the original Juliet study by Novartis in diffuse large B-cell lymphoma in 2016. Our first patients were enrolled. And then it basically took us several years for that data to mature and eventually lead to approval of tisagen lec-lucel, which is otherwise commercially known as Kymriah from Novartis, and it has been made available for our patients and now approved by our TGA and the gene technology regulator for use. And now at my centre, which was only one of only 2 hospitals in Australia that were approved by our national government to administer CAR Ts, we have now provided that free of charge to over 100 patients for diffuse large B-cell lymphoma, other lymphomas now, and of course, acute B-cell lymphoblastic leukemia. The pipeline is extraordinary because although CAR T-cells dominate GMO-based cell therapies worldwide, the breakdown showing the development is extraordinary. These are CAR T-cell based therapies, but they can also involve NK cells in terms of development, or indeed T-cell receptor T-cells or other based therapeutics. But moving forward, the predominant application of these CAR T cells are in oncology. And it is predicted that that will only increase because look here on the left-hand pie chart, gene-based therapies are 20% approved right now, but the pipeline of development suggests that going forward, that will more than double to 55% of all of the different technologies available. In Australia, we have a number of centres that are able to administer these gene and cell-based therapeutics. And we also have some national collaborative scheme, national biologic genes in small pharmaceuticals, and most recently in the newest one, cell and gene-based therapeutics, and Royal Prince Alfred Hospital is the centre that's available. So if I've got another 5 minutes, I will just cover some of the examples that I wanted to bring with you that I've spoken about in this room over the past 10 years and highlight some of the clinical trials that I've been involved in pursuing cell and gene-based things. And really I started in this area back in the early 2000s with hemophilia. As a hematologist, hemophilia was one of the most compelling clinical challenges that I wanted to address in my career, and I've been humbled to be able to bring that to realization. And so we were the only centre in the Southern Hemisphere to be involved in adeno-associated virus gene therapies that were published in 2 reports in 2006 and 2007 regarding factor IX based gene therapy or hemophilia B. And now more recently, we've had publications describing success in hemophilia A and hemophilia B in 2017 and 2021. And I'm happy to be able to report that in the European Union, BioMarin, a company that has been pursuing gene therapy for many years, has had an approval for hemophilia A only last month. In parallel, we have participated in the Bluebird Bio beta thalassemia clinical trials that was published in 2018. And the European Union conditionally approved that therapy in 2019, but the company BioMarin withdrew the therapy about 18 months later because they couldn't sell it to anyone because it was so expensive, nearly €2 million per patient, although it is known to be curative in transfusion-dependent non-beta 0, beta 0, beta thalassemia. And more recently, though, this technology from Bluebird Bio has now been approved again last month by the FDA in the USA. So it was withdrawn from commercial use in Europe after first approval in 2019, but now they have pursued the US market and have just received approval after 2 years of delay. I mentioned Kymriah and Yescarta from Kite Gilead, which was approved in Australia in June 2020, and we have been administering that ever since. And finally, and I will mention briefly, the use of induced pluripotent stem cells, which I know is close to Prof. Ong's heart in terms of our clinical trial in graft versus host disease, published in Nature Medicine in 2020. So very, very briefly, as clinical examples, in hemophilia, all of you know that it's a bleeding disorder. A and B are clinically indistinguishable, but most patients these days receive recombinant or plasma-derived concentrates, which is episodically administered. And that sawtooth pattern at the bottom of the graph there shows the rise and fall of the levels of factor A, 8 or factor 9 in the blood of individuals respectively. But people with severe hemophilia are less than 1% of normal. Hopefully most people in this room are between 50 and 150%, more or less 100% of the normal level to protect us all against bleeding. And so back in the year 2000, we administered gene therapy to a couple of patients as part of an international trial led by the Children's Hospital of Philadelphia and Kathy High, and they were published in Nature Medicine. And I don't know whether this is going to project or present, but it's a brief video. I don't know whether, is there sound? Is that going to work? Sorry, I should have talked about that, Kim. Is it possible to get that? You got it? You got it? You got it? Maybe not. Doesn't matter. It's not. That's okay. Never mind. Never mind. That's okay. That's okay. So basically, this is. Don't worry too much. I will take that off and move on to, I anticipate, the slide's not progressing. There we go. So basically, this is the response to a single injection of AAV-based gene therapy into a peripheral vein of individuals with severe hemophilia. In this case, this is factor 9, hemophilia B. And as you can see, these patients all received a clinical benefit, and that was presented first in 2016 and then subsequently published in 2017 in the New England Journal of Medicine. Some of the patients suffered an immune response to the coat protein of the viral vector AAV, which is still one of the hurdles that we need to overcome, one that I'm actively involved in studying in my research lab. But steroids and steroid-sparing agents are the mainstay of managing that challenge. And in the end, it leads to the successful therapy for hemophilia A or hemophilia B. And those reports are now established. And as you've just heard, now has just been approved in the United States of America in the case of, in the EU in the case of BioMarin for hemophilia A. And the anticipation is that there will be several companies all competing, Fruitline, Unicure, Pfizer, Spark, and other companies all competing for approvals in the United States of America and large markets. In the case of thalassemia, many of you know this disease, and I know some outstanding professors from UTAR and the affiliated hospitals study and provide service in thalassemia. And I've been involved in the gene therapy using ex vivo gene-directed therapy in CD34 hematopoietic stem cells in the context of a myeloablative bone marrow transplant using autologous cells with a version of beta globin introduced into these cells, which is capable of curing individuals with transfusion-dependent non-beta 0, beta thalassemia. And this is one of my patients, and I love this photograph because that was now 4 years ago when he received his last blood transfusion prior to his bone marrow transplant with gene-modified autologous hematopoietic stem cells. And the last example I will give, because Prof. Ong and I were talking about it, is our pursuit of induced pluripotent stem cells and their derivative cells as a foundational technology to be able to use all cells potentially in the body, because they derive from iPSCs or embryonic stem cells. And so the disease that we've partnered with a company called Cynata to address is called acute graft versus host disease. Now, many of you know that allogeneic bone marrow transplantation is one of the foundational cures of acute myeloid leukemia and other acute leukemias, and indeed some other cancers, because of its graft versus leukemia and graft versus tumor effect. But one of the most feared consequences of allogeneic bone marrow transplantation is graft versus host disease, which can be life-threatening. And indeed, if you don't respond to steroids, then you can either have one of or combinations of gastroenterological, liver, or skin-based damage, which leads to a horrible quality of life and sometimes death, often associated with infection. And it's probably contributes directly to about 10 to 15% of deaths following allogeneic bone marrow transplantation. And so it's a major unmet need, and only ruxolitinib, the JAK2 kinase inhibitor, is available for therapeutic use. So this is an unmet medical need, and it turns out that mesenchymal stromal cells have been examined for now nearly 20 years in the context of acute steroid-resistant graft versus host disease. And they have a number of different mechanisms of action that are plausibly able to address this serious disease. And this is a paper that I published with Killian Kelly, basically describing the mechanisms of action of mesenchymal stromal cells in graft versus host disease, and many of them related to IDO, the enzyme IDO, but they are listed here and detailed in a little bit more than time is allowed. But basically in 2004, Katerina LeBlanc published a paper in The Lancet describing the use of mesenchymal stromal cells in steroid-resistant acute graft versus host disease that showed dramatic benefits. And as a consequence of that, the world has been pursuing the possibility of using mesenchymal stromal cells in graft versus host disease. And I can't believe it, but when we got to review all of these studies, there are some 60 published studies worldwide using mesenchymal stromal cells in graft versus host disease, but they are of all types. Some of them using different cells from allogeneic, autologous, or matched unrelated or matched related individuals. Some of them have been in the context of using cells from different sources, some different phases of trials, some in adults, some in children, and so on, and some in acute graft versus host disease or combinations of chronic. And so there have been a lot of reports, but nothing conclusive. And so really the challenge has been standardizing the use of MSCs in clinical development so that we can make sense of the results. And the problem is that we know that over time, as we grow MSCs ex vivo, they can change their potency, they can change their biological activity, they senesce eventually, and they decrease in efficacy in vitro surrogate assays. And so this limits the number of doses that you can give of MSCs as you expand them ex vivo. And so you require new donors, and it creates more variability. So the idea of using induced pluripotent stem cells that won the Nobel Prize to Shinya Yamanaka and Sir John Gurdon only less than a decade ago is that they can be an essentially unlimited source of providing for mesenchymal stromal cells in this case. And the comparison of the different types of origin of cells that can give rise to MSCs is shown here. And so I would put it to you that iPSCs present an ideal starting material, and the challenge is how to grow them in an industrial scale. And that challenge was overcome by colleagues at the University of Wisconsin. Jamie Thompson is one of the discoverers of human iPSCs, and his colleague Igor Slukovin, who's a colleague and friend, who identified that you needed to pass the cells through an intermediate stage called mesenchymoangioblast stage of differentiation, which I won't go into the details of, but essentially this is a comparison at the top of the conventional way that we grow seeds from origin in bone marrow or even in fat tissue derived MSCs, compared to the Cynata process, which requires this intermediate stage of mesenchymal angioblast development and then cloning so that it guarantees that there are no iPSCs in the therapeutic drug product that would otherwise potentially lead to tumors recipients. And so these allogeneic iPSC-derived MSCs are used in the treatment of GVHD in a clinical trial. It's called Cymerus and essentially was granted orphan drug designation in March 18, after which we started our clinical trial. It's supplied in liquid nitrogen, a vapor phase, and there's 10 to the 8 MSCs per 20 ml infusion, and it's just administered IVI. The issue here is that it's entirely scalable. And at the current scale, the manufacturing that was undertaken by the company, Cynata, was one single donor was able to give 19 million clinical doses, more than we require for any future use. And so that process is even more scalable beyond that amount, quite in a straightforward manner. So then the clinical results I'll rush through, but basically it was 2 doses infused at day 0 and day 7. And essentially, it was a safety and tolerability study that was performed in the UK and in Australia, 7 clinical sites, as shown here. Royal Prince Alfred Hospital, I was the international coordinating and principal investigator, and there were the other centres in the UK and Adelaide. And essentially, the results showed that these infusions were safe as was anticipated. There were 5 treatment emergent adverse events in 4 participants that you can see listed there: abdominal pain, diarrhea, and neutropenia, but no treatment of emergent adverse events were assessed as probably or definitely related to the drug product, which is called SIP-001. And essentially the summary is that it was safe and well tolerated, and remarkably, we saw an observed clinical response rate of over 50% and an overall survival rate of 86.7% at day 100, which is as good as the best therapy worldwide of any type in acute steroid-resistant graft-versus-host disease, and that was published and highlighted on the cover of Nature Medicine. The results are summarized here. These are the swimmer plots on the right-hand side of the patients that were enrolled, 14 patients, and you can see that the grade of graft-versus-host disease, the majority of patients started at grade 3. It only goes up to grade 4, which is very, very severe. But the vast majority of the patients saw a substantial improvement in their clinical grade following infusion of the MSCs. And that's prompted then the design of a new study, which will be commencing later this year, a phase 2 study of this promising mesenchymal stromal cell derived from iPSC drug product, which is to use in combination with steroids at the diagnosis of acute steroid-resistant graft-versus-host disease. So basically, in summary, we've developed a number of different technologies which I've highlighted and spoken about in terms of the interface between regulatory and clinical development. These are the publications that have highlighted some of these developments in hemophilia, in thalassemia, and more recently in graft-versus-host disease using the iPSC-derived cells. And all of these have converged on the possibility of using a gene-based and cell-based therapies to improve human health. I don't think we get rid of drugs tomorrow, but hopefully we'll be able to interface and interplay cell and gene therapies as having a serious and useful contribution to human health. As always, I want to thank my group. This is from a slide taken now a few years ago, because we haven't had a chance to go on a retreat without masks, but this group is now still very happy and functioning very well. These are the funding bodies that support some of the research that I do, and I want to apologize for going a little bit over time with our technical challenges, as well as starting a bit late. I'm very happy to answer questions. Congratulate UTAR for a wonderful 20 years and to have many, many decades more of great success. And I hope I will be able to continue to visit you going forward. Thank you very much.
Thank you very much, Professor Rasko. It's always a pleasure to be bringing up the latest development and exciting news on cell and gene therapies. Now the session is open up for Q&A session. For the participants who are on Zoom, you may type in your question in the chat box more to and more myself. And for the just raise your hand and you have extra points.
Thank you for the
Great question. So the question was, if you want to do gene therapy clinical trials in Australia, you need to do small or big animal clinical trials in animals prior to commencing. And the answer is it depends on what other studies have been done worldwide. And essentially, one starts a dialogue with the regulators. As a general proposition, you would need to show proof of principle in an animal model prior to proceeding with human studies. So obviously you can't just do anything you want without any evidence. But if, for example, there has been a pivotal study done in another center that has either been peer reviewed or a regulator has reviewed it in another center, or for example, even a drug has been approved elsewhere under a jurisdiction that is recognized within Australia, then it's quite possible to say, look, we're going to use that file of evidence to support the fact that we should be allowed to proceed. But if this is an entirely new, completely novel way of treating some disease that you want to use in humans, then yes, it is required that you need to use what is called an appropriate clinical model of relevance to humans. So for example, if it has an immune context to it, one would probably think that a large non-human primate model might be required. But if it is something that doesn't get complicated by that kind of variability, relevance to human context, then maybe animal model might be appropriate. Regulators, yeah. Prof. Vera.
Thanks for the lecture. And going back to regulations, and we talked about national regulations, how far do you think global regulations are needed as far as GMOs are concerned? I think we've seen that with agricultural products where only certain companies hold patents, and we really have our entire food crops dependent on these few companies. So getting global reach and global accessibility, not only to just to food crops, but also now the modality interventions. How do you think the global community has to get involved in order to make sure that this is
Yeah. Thanks, Prof. Vera. So the question, just to repeat, was essentially how far should we be able to go with global regulatory consistency and in terms of GMOs. And, you know, you highlight an extremely important and difficult challenge, which the International Society for Cell and Gene Therapy and other organizations should be well equipped to promote, but it has almost unbelievable difficulties in pursuing. The reasons why we should do this are absolutely compelling. The companies that are pursuing gene technologies are so frustrated at the additional costs in addressing regulators within each country that it's not just the challenges of suiting the gene technology to individual ethnicities or races or countries and laws and regulations, but it's also a matter of complying with social expectations and what the individuals in the country want. But that said, we think that 80% at least of the regulatory challenges are consistent within each country. And so the idea would be that even, let me particularize it to my center. We have more than a dozen clinical trials open at any given time, sometimes 20 different clinical trials. Every single company that comes to us to want to do a clinical trial has to have its own specific regulatory compliance, its own specific, and we know that 80% of everything is the same between each of the companies, and yet they all have different regulatory requirements. They all have different impositions on our manufacturing site. And you know, we say within our department, why can't you all just kind of agree on a set of standards? So the companies are responsible, but international community is also responsible. If we are able to achieve some harmonization, and that's the word, through the international community in terms of regulatory compliance, that would be a major improvement in terms of allowing our patients to get earlier access and just as safe access to some of these life-saving technologies. So I agree with you that regulatory harmonization is a major impediment to accelerating the development of these technologies. But if you just reflect on even Europe as a place where you say, okay, there's the EU, the European Union, they all agree that they are part of a union. It's wonderful, isn't it? They're all harmonized, they're all happy together. In fact, when companies go to Europe to bring their gene technology, what they find is they have to kind of go through all of the regulatory hurdles of the EU. That's very good. Thank you. You've now passed. Congratulations. And then any country that they want to go to within the EU, they have to comply with the individual countries' regulations as well. It's madness. So what you would think is harmonization is just another layer on top of regulatory obstruction. And so I'm only saying I agree with you. It's a major challenge, and I've offered no solution whatsoever other than to say, yes, we should be doing a lot better. It's a big issue, and that's why we need regulators to come together internationally at big international meetings through the WHO, through international harmonization, and try and achieve some level of harmony so we can, progress this this hurdle. A couple of questions.
Good morning, Prof.
Good morning.
I'm glad that I'll get to see you in person finally.
I have two questions. First one is, what is your biggest challenge in getting challenges from fresh? So the second one is, as you said that the regulation about but if you go back, what does that mean for challenges therapy development?
Thank you.
Thank you.
I guess the first question is about
personal, and I think I could speak for ages about that, but I think funding is really one of the biggest hurdles that I faced in my career. And my solution was, of course, to do my best to apply as widely for grants as possible and employ the best possible people that I could to do research, but also partner with industry. Because for my career, I have developed clinical trials, both in-house, but also in partnership with industry. And that has facilitated infrastructure, knowledge, and regulatory compliance so that we've been able to pursue some of the expensive investigator-initiated studies that we have, been able to enjoy some success in. So I think you have to, and we are all well-educated, we all have ideas, we all want to pursue new things that we think are important, but unless one can convince the funders, whether they be philanthropy, government, international funding, or whatever it is, mum and dad, whatever it is, we have to be able to source a reliable source of funding in order to pursue our research. And that's something that I paid a lot of attention to over my career whilst trying to do the best possible science I could. You asked me a provocative question, which is it good to delay or accelerate the promulgation of laws related to regulation? And, I guess you're asking the wrong person, because I really have devoted my whole career a rigorous independent regulatory controls of gene technology, because it's the only way that I think you can establish a fair and a robust environment where we know that the results of clinical studies, and we know the safety profile of these new medicines that scare many members of the public, we know that they are safe to proceed. But I have, so I am very strongly in favor of bringing laws and acts and regulations into effect. But I'll say that I'll answer it in a different way. I'll answer it in the way that it is only good for Malaysia. If you do not have laws and regulations that allow big multinational companies from Europe, from America, from Australia, and from Asia, if you do not have laws that they can come to and say, look, the clinical trial that was done in Malaysia is as good as any clinical trial worldwide because their regulations are just like ours. And therefore they did the same study that we would have done. And they can be published and they can be recognized internationally as robust and well regulated. Unless you have that regulatory arm's length government imposed environment, you can't come to the table of international companies or the table of international regulators and say, look, our study is robust. We have done everything independent, everything at arm's length, compliant with laws that are consistent with international harmonization, going back to Prof. Vera's question. So I think the reason why you want to have laws and regulations in Malaysia is so that you can play on the international field in an equal way. It's a good question. One question over there, if there's time. Morning.
Like you said, like the last therapy is such a nice example. So definitely the global regulatory body will be a bit difficult because of the personalized use using patients themselves.
Yes.
It's difficult to just on a global scale to look at that personal level of compliance. But the success of the Comirnaty Pfizer vaccine, that is gene-based therapy. And of course, because of its success, it's so clear the use of cell-based gene-based therapy. So my question is, how much gene-based therapy can we convert, replace cell-based therapy? Then you say in the slide that we're going to be more gene-based therapy. So if you could let us know, I'm sure you've done your method of cell gene-based. So how can we replace our cell-based with gene-based so that it's easier to
yeah. I think I understand where you're going, and you know, it's very easy to give you a short answer, which is in vivo gene modification. So in other words, we now talk about two different types of gene therapy, in vivo and ex vivo, where, for example, with the success of the CAR Ts, we take the cells out of the body, autologous cells, genetically modify them and put them in. And there's a very robust manufacturing process. And you're right, there's variability because the actual cells are different for each individual patient. Ultimately, every company that is doing CAR T cell therapies wants one thing, and that's off-the-shelf technologies. They certainly don't want to be pursuing autologous cell therapies. There's a pain to have to take somebody's cells, ship them to a manufacturing place, either centrally or someone else in the world, locally or elsewhere, manufacture them consistently with very specific criteria, and then return them to the individual patient. That's unbelievable logistics, cold chain maintenance, requirements for regulation. It's a nightmare for those, and it's unbelievable that they have succeeded in doing that. But ultimately, the trajectory for all of these technologies off the shelf, in other words, iPSC-derived technologies or matched technologies where you can just have a cell in, you have leukemia, fine, here it is on the shelf, put it back in your body. So iPSC-based cell therapies is one way to make things more accessible and reduce the requirements for autologous-based therapies. But in answer to your question, how do we convert, how do we get rid of cell-based therapies and replace them with gene-based therapies? Then it's all in vivo. So the proposition, so a lot of the companies now are pursuing iPSC-derived NK CARs or T-cell CARs or all of the different iPSC-derived therapeutics using off-the-shelf cells. But the actual trajectory is not to do any of that. And it is exactly as I think you're asking, to use gene-based modification in vivo. Don't waste your time taking any cells out of the body. Don't waste your time using iPSCs off the shelf. Inject, if you are clever enough to get a technology to be able to be injected in vivo, and it targets the very cells that you want to genetically modify with CRISPR-Cas9, which can be a permanent change in those cells, you can make in vivo T cells, CAR T cells, that are now armed and programmed in your body to target the leukemia or the lymphoma or another solid cancer. So potentially, if we can harness the power of gene editing, not just CRISPR-Cas9, but zinc finger nucleases, meganucleases, things that have been around now for 15, 20 years and are approaching, very much approaching clinical use in lipoprotein diseases and so on, these technologies can be used in vivo. And the basis for that is the AAV platform, because it's become now the safest technology that we have for delivering genes in vivo, as well as marry that now with the new liposomal-based technologies and so on. So I think your question is really well put and gives me the opportunity to talk about where we might be in 10 years or 15 years or 20 years time, when you basically don't need to take any cells out of the body. You just inject the gene and you get a genetic modification in vivo. That's where I think the future is. Yeah, thanks, Dr. Lee. We do.
Thank you very much again.